Hello everyone, and welcome to EssenBioScience's webinar series. Today's topic will be NeuroTrack, label-free, fully kinetic neurite outgrowth assays in human ES-derived neurons. And in a minute, we'll pass things over to our speaker today, Dr. Del Trezise, Director and Site Head at EssenBioScience Limited in the UK. Our presentation today will last approximately 30 to 35 minutes or so, and will be followed by a question and answer session. So without further ado, I'd like to turn things over to Dr. Del Trezise. So take it away, Del.
Okay, good morning, good afternoon, good evening to everybody, wherever you may be in the world. It's great to see so many people online and interested in NeuroTrack today. As Jason mentioned, the title of the presentation today is NeuroTrack, label-free, fully kinetic neurite outgrowth assays in human iPS-derived neurons. The structure of the webinar, which I anticipate will take 35 to 40 minutes or so, is as follows. I will give a brief introduction to live cell imaging and neurite dynamics. We'll then follow that with an explanation of the NeuroTrack methodology workflow and some metrics and pharmacological validation. Then walk you through a case example where we've used the NeuroTrack approach to study the formation and stabilization of neurite networks in human iPSC-derived neurons, and then we'll conclude with a short wrap-up at the end. So, apologies for those of you that may be already familiar with the concept, but live content imaging may be something that is new to many of you. So here is our definition of live content imaging. So just to go through this slowly, live content imaging is the acquisition, analysis, and quantification of images from living cells, and that's a key point, that remain unperturbed by the detection method, and that's another key point, allowing for repeated measures over long periods of time, from days to weeks, and that's another key point. So together, for monitoring phenotypic biological events that unfold over hours, days, and potentially weeks, we see that live content imaging is an extremely powerful approach. The key features of live content imaging technology are outlined on this schematic. Importantly, it shares some of the good attributes of high content imaging which you may be more familiar with. You can use fluorescent probes, you can do quantitative plate-based pharmacology through image analysis, you can multiplex more than one fluorescent channel. Unlike most high content imaging approaches, we use light microscopy and proprietary high definition phase contrast optics to do a lot of assays in a label-free way, so without fluorophores, and NeuroTrack is an example of that, which we'll come to in a second. All of our assays are done from truly live cells, where we take repeated measures of the phenotypic parameter of interest over time and fully resolve spatiotemporal events over the course of days and where of relevance and interest over weeks. This is all put together in a way that's what we call plate reader simplicity, with a graphical user interface which is very intuitive, some powerful but straightforward analysis and software tools. The premise here being that this is imaging for people that might not necessarily be experts with imaging and image analysis, are still able to assemble these assays, gather meaningful data, analyze and interpret it. So hopefully that's clear. EssenBioScience has a platform which is specifically designed to do these types of experiments. It's called IncuSight Zoom. It's a mature technology which has been around for several years now. The latest device was upgraded and launched about a year ago. As listed on this slide, it's a fully automatic two-color phase contrast and fluorescence microscope that resides within any standard tissue culture incubator. It stays there largely for the duration of its life. You take it out once a year for service and maintenance. It images live cells kinetically, and through the user interface that I referred to, it allows you to gather images and to analyze and quantify those images and turn those images into data, and that data into knowledge and conclusions. As you'll see, the InkiSight has positions to house cells plated on either tea flasks or petri dishes or microtiter plates. A single InkiSight can house up to, for example, 6 384 well plates or 6 96 well plates, or any permutation and combination of plates and flasks that you so choose. You simply prepare your cell plates, add whatever treatment conditions you would like, place them in the InkiSight, close the drawer, close the door of the incubator, program the software, and off you go. So the cells stay still. There's a single microscope objective that resides underneath the cells, which moves around, focuses on particular fields of view or points of interest within the microtiter plate, and then moves on, having taken an image, to the neighboring well. So hopefully that's reasonably clear. So let's move on to just consider for a moment neurite dynamics and in vitro neurite outgrowth assays. As I'm sure you're well aware, that neurites are formed through a development process which is in itself very dynamic. Neurites are formed, they retract, sometimes they are cropped, sometimes they regrow when nerves are damaged, sometimes they develop and are strengthened through synaptic connections. Importantly, the biology of neurite outgrowth in vitro models shares sufficient parallels to the same biology in vivo for such models to be considered useful test systems for exploring the type of things listed on the right hand side of this slide, so which includes neurophysiology measurements, the processes of neurodevelopment, disease models, testing samples for neurotoxicity, for example, as well as a range of other things. Until very recently, or until the last few years I should say, almost exclusively this work had been done with neurons obtained from experimental animals, since it was next to impossible to obtain healthy, intact neurons from humans. The promise of stem cell technology has begun to change this, and now we live in a very exciting world for anybody in the neuroscience field, where it's possible to take somatic cells from patients, to reprogram and differentiate those cells into specialized, in this case, neurons, and then to begin using those human-derived neurons as a disease model system. And importantly, those human-derived neurons retain the genotype, and there's sufficient evidence to suggest they also maintain some of the phenotype and genotype of the patient from which the somatic cells were isolated. And this is a very exciting development in the field currently, and I just have taken this from a review article of a number of papers in which a disease phenocopy has been illustrated in iPSC-derived neurons taken from patients with particular genetic mutations. Now, I should caveat, this logic with the recognition that these cell models are far from perfect. There's a long way to go before this combination of stem cell technology and fully validated and understood neuronal models is accepted, but nevertheless this is a work in progress, and we see through live content imaging that we can add substantially to the development and the validation of these neuroscience disease models. Okay, so let's come on now to NeuroTrack workflow and methodology. It's really very simple, as I alluded to from the previous slide. You can take pretty much any neuronal type. We've done work with immortalized neurons and neuronal cell lines, primary neurons including cortex, striatum, hippocampus, midbrain from mostly rats, the work that we've done, or iPSC-derived neurons, and we've worked predominantly with eye-cell neurons from CDI. You take those cells in suspension and then you plate them onto a microtiter plate. This can be either a 96 or a 384 well plate. You can include a plating substrate in laminin or poly-D-lysine as needed. You allow the cells to adhere for a while, and then you can treat those cells with any test samples, whether it's small molecules or antibodies or genetic manipulations, on the plate. You then place the plate within the inky site and schedule the inky site to gather the phase contrast images. And you can do this as frequently as you wish. Typically for long-term monitoring, we would take an image, and sometimes more than one image per well, perhaps every 2 to 6 hours, and measure for anywhere between 3 to 15 days, depending on the biology that we were monitoring. This next slide, it's quite hard to see the very fine neurons of these rat cortical neurons, but this is intended to explain how we mask these phase images to turn images into data. So on the upper part of this slide, we have 3 images taken at 24 hours, 72 hours, and 120 hours of rat primary cortical neurons plated on poly-D-lysine, beneath which we have the same images overlaid upon which is the inky site NeuroTrack imaging mask. And in this case, it's labeled the cell body clusters, we call them, in pink. So anything which is round and has the morphological properties of a cell body is categorized as pink and segmented as such. And anything which is long and thin and spidery and bifurcating and of neurite morphology is labeled or segmented in the pale blue. So you can see clearly here, as a function of time, these cells send out neurite projections and develop after 5 days a fair elaborate neurite network. It's a little easier to see that if I show you a movie, so let's just jump out of the PowerPoint for a second. So just a reminder, for those of you who can see my mouse here, if you'd like to just select your maximal resolution to see this movie as best as you can, the highest number here, I'm 720p. These are now human eye cell neurons plated for 1 hour, and as you can see, even after an hour, there's some short neurite projections from these cell bodies. Most of the cells are separated from each other, but some are clustered together. I'll now play this movie. And what you'll see, so these images are taken 2 frames per second, they're displayed at 2 frames per second, 6-hour intervals, and the total length of this film is 8 days. So you can see that the neurites really become quite elaborate really quite quickly. This film then loops back to the start, and we can see where we start from the beginning. So you can see the quality of the HD phase imaging, and the by the naked eye you can see pretty much all of these neurites in the field of view. If I now show you the corresponding movie for the inky site mask, so in this movie the cell clusters are labeled in pink and the neurites labeled in yellow. So here we go, and we can see that the NeuroTrack algorithm is pretty good at recognizing each of these structures. It's not perfect. You'll see in the movies the odd neurite that it perhaps doesn't detect, or the odd cell body cluster that it doesn't detect. But for the most part, the ability of the module to discriminate and discern the relevant structures and quantify them from the phase image is pretty useful. So hopefully you now get an idea how we can take that image, convert it into data, and turn that data into screening information or assay information. Okay. So back to the PowerPoint. So just one final point before I show you some data examples. The metrics that we quantify or that we gather from this image analysis are summarized on this slide. Total neurite length in the field of view. So we don't measure individual neurite length. If there are 50 neurons in the field of view, we sum all of their total neurites to generate a value in millimeters per millimeter squared. We can count and identify the area of the cell body clusters, and we can quantify the number of bifurcations, the number of branch points for the total number of neurons in the field of view. And then we can reference some metrics to others for normalization purposes. So for example, we can calculate the neurite length per cell body cluster count, or the neurite length per cell body cluster area as examples. I'll come to dovetailing this with fluorescence data later on in the talk. Okay, so let's get into some data examples. So this first slide summarizes data from 3 different cell types: some Neuro2A immortalized neurons, some rat cortical neurons in the red, and the human eye cell iPS-derived neurons in the gray. In the left-hand panel is time course of the neurite length expressed in millimeters per millimeter squared. This is 20x imaging, taking 2 images per well and a 96 well plate. On the right-hand graphic is the number of branch points as a function of time for the same cells. What's obviously striking from this is that each cell type displays its own characteristic profile of neurite formation and neurite outgrowth under these conditions. The eye cell neurons are certainly the fastest at developing their neurite network. The Neuro2As really take several days. These are treated with their transretinoic acid. They take several days before they really develop the elaborate neurite network that you see, for example, in the cortical cells. So that's probably the simplest example of a data set. Moving on now to show you the normalization per cell body cluster. So in this example, this is data with human iPSC derived neurons, eye cell neurons from CDI, plated at different cell densities ranging between 80 to 25,000 cells per well, and their time course again of their neurite length on the left, and neurite length normalized to the number of cell body clusters in the field of view on the right. And you can see how obviously the more densely you plate the cells, the greater the total neurite length, which is unsurprising. Also, the greater the number of cell body clusters. And if you normalize for cell body cluster, for the most part, these analyses then overlay. So this is a reasonably helpful normalization process. It doesn't always follow this perfectly, since if you overload or really sparsely seed the cells, then it does have a bearing on their ability to form and sustain their neurite projections. Hopefully that's clear. Okay, so just to take one step back, you cannot, or at least we cannot, do this imaging and quantification on any type of microtiter plate. We've worked very hard to look through and to assess the quality of a wide number of different plates. The key thing is that the optical clarity and quality of these plates needs to be really good for the IncuSight and NeuroTrack algorithm not to confuse scratches, for example, as neurites. Our preferred plate is generated by a company called Technoplastic Products, TEP, catalog number 92696 as a 96 well plate. In our experience, these are the best optical resolution of any plate that we've tested. They're not premium priced. They're very reasonably priced. So there's no expensive consumables in this assay other than the cells. To be fair, if you use IPS cells, they're still relatively costly. In this slide, it's hard even to see by eye the scratch in the competitor manufacturer plate on the left-hand image, shown by the orange arrow. But you can see in the corresponding image, the third from the left, that the IncuSight algorithm, the NeuroTrack algorithm, sees that scratch as a neurite, and it quantifies and therefore overestimates the total neurite length in that field of view. The other issues that you will find with different plate manufacturers are that striations in the plate can impact of this phase algorithm to work. We've also found that we believe a glue-related artifact with certain plates. Neurons are very sensitive to any chemistry around them, and certain plates, we found them to be toxic to the neurons, and we believe that's related to the adhesives that are used to assemble the plates. Any plate with a rough texture or scratches or imperfections is not suitable for this type of imaging. But in our experience, and we've been doing these things for quite some time now, the TPP plates are of good quality, they're reliable. We've not had any batch-to-batch issues, so we would recommend that you use these if and when you're doing these types of experiments. Okay, so let's just turn that kind of imaging and data now more into a screening world for a moment. This is a InkeSight GUI view, 96-well plate view of a exemplar experiment. And this is a relatively uninteresting experiment in many respects, in that all we've done is simply plated rat cortical neurons, E18 cortical neurons, on poly-D-lysine in every single well of a 96-well plate, and then monitored their neurite formation as a function of time. In each well here, the x-axis is 0 to 5 days, and the y-axis is 0 to 0.4 millimeters per millimeter squared of neurite length normalized for cell body cluster. The reason for showing you this image is just to give you a feel for how the InkeSight assembles thousands and thousands of images and processes into something which is reasonably easy to digest and interpret, and gives you an idea of the well-to-well consistency. Typically in our hands we have plate CVs in the region of about 7% or 96-well assays, which is perfectly good for most of the questions that we wish to ask. You can also do this 384-well plates. It's not quite so good. The imaging gets a little more challenging as you further miniaturize. Our typical plate CVs, however, are not that bad. About 12% is fine. Our driver for 384-well assays is not so much throughput as a desire to maximize the use of quite expensive cells. So you need fewer cells, you get more data per cell, if that makes sense, if you miniaturize to 384. And when we're dealing with some of these IPS-derived cells that we purchase, then it becomes an important ability to miniaturize these to 384. So certainly possible to miniaturize these assays to 384. Okay, so just before we move on to the case example, just show you a bit of validation pharmacology, really geared to exemplify the power and the value of these kinetic measurements. So in this particular series of experiments, we've taken a actin polymerization inhibitor, cytoclasin D, and studied across a range of concentrations the effect of this molecule on the neurite outgrowth in panel A, rat cortical neurons, panel B, human IPS-derived neurons, and panel C, Neuro2A immortalized cells. And what's very striking in comparison to the red controls, the vehicle-treated controls in each cell type, is the differential, temporal, and absolute profile of the effects of cytoclasin D. So in the Neuro2A cells, the molecule simply prevents neurite outgrowth at the 3 concentrations that we tested. In polar opposite contrast to that, in the human IPS neurons, what we saw at the 2 highest concentration was an augmentation of the total neurites per cell body cluster. Most interestingly is the profile we saw in rat cortical neurons, which is at the higher concentrations we saw an augmentation of the neurite formation over the first 30 hours. If you took an arbitrary endpoint measure at 30 hours, you'd have concluded that nothing happened. If you measured it through to 60 hours, you find that those higher concentrations actually inhibit neurite outgrowth. So there's this temporal, concentration dependent and differential effect on different cell lines of this particular compound. And this is part of the value statement for this temporal imaging, which is really, really difficult to generate this type of data using high content imaging, for example. You will of course recognize that any type of cell death within these assays is a consideration and complexity that one needs to be aware of. There are ways in which we can duplex these phase imaging measurements with fluorescent probes. I illustrate one example on this slide, whereby we use a cell-impermeant DNA binding fluoroprobe. This is data with YoPro3, but you can equally well do it with DRAC5, 7, or even some green fluorescent labels. These are what we call mix and monitor. So you simply add them to your cells and your media serum. If the cells are healthy, the fluoroprobe cannot access or traverse the cell membrane, and you see no fluorescence. If the cells begin to become unhealthy and their membrane is compromised, the probe enters into the cell, combine the DNA in the nucleus, and light that up, which can be detected in the red channel. So the three graphs on here, left to right, are the neurite length per cell body cluster of control cells in the red, and in the present, this case, of a protein kinase C inhibitor, row 3, 1, 8, 220. And you can see clear concentration-dependent inhibition of the total number of neurites per cell body cluster in this experiment. This, however, is associated with a clear induction of cell death, as monitored by the red object counts of the total number of nuclei appearing in these images as a function of time. You can analyze these time courses in several ways. On the right-hand side is an area under the curve analysis, showing the concentration response curves for neurite length in the grey and cell death in the red. Just one more slide on this topic. This shows, perhaps explains it a little more clearly, the segmentation mask on the left and some of the phase and fluorescent blended images on the right for cells in the presence of YoPro3 and a cytotoxic agent, where the blue outlines of the segmentation mask for the cell body clusters. The yellow, looks almost white on screen, is the segmentation mask for the neurites. And then the red spots are the nuclei of these cells in the field of view. And blown up on the image on the right, you can see one very clearly dead cell, and then the emergence of red nuclei and a number of other slightly healthier looking cells at this time point. So hopefully that's clear how you can duplex neurite outgrowth and neurotoxicity. You can do the same with some caspase substrates to duplex neurite outgrowth with apoptosis. Okay, so let's come on to a case example quickly. In these series of experiments, what we set out to do was to assemble assays for dissecting the differential biology of neurite formation and network stabilization. We figured that this would be interesting to do. It's not something that's readily accessible by endpoint measures. And our approach was to take human iPSC-derived eye cell neurons. This work was done on 384-well plates with on-plate quadruplicates in neurobasal media with glutamax and B27. And we took images every 2 hours for up to 14 days and calculated a range of metrics. The data I'm going to show you is with total neurite length. There were no refeeds in this experiment. And the treatment groups that we made were to look at the effect of excitotoxic agents in the absence and presence of different modulators at t equals 0, so before the neurites had formed, to see if we could modulate or inhibit the process of neurite formation, and then do a parallel set of experiments where we waited for the neurites to form and stabilize their network to begin with, and then treated those cells with excitotoxics in the absence and presence of other modulators. Here is the first data example here. So on the upper panel here is now data where glutamate was added at t equals 0, the network formation assay, and then underneath the network disruption assay. The red in both cases is the control treatment, and then the darker and darker colors are the presence of increasing concentrations of glutamate. And somewhat to our surprise, what we saw was that if you add glutamate at the start of these experiments, it has no effect on the ability of these cells to generate neurites, but the stability of that neural network is compromised. And over the long haul, these neurons don't fare well, and their neurites don't stay as stable as you see in the controls. And if you quantify different epochs, you know, early, late phase, then you can see from the concentration response curve this clear inhibition of the late phase without any effect on the early phase, shown by the grey symbols. If you wait for the network to develop and then add the glutamate after 3 days, glutamate again doesn't do anything profound immediately, but it takes a few days before the network begins to degrade, and you can quantify this. Just as a reminder, behind all of these data points and these time courses is a movie. So I'll just show you a control movie. So this is 10x imaging in 384, so not quite as precise as I showed you in 96. So these are the control neurites. It's quite a large cluster of neurons in the middle here. You can see they send out these neurite projections, and they're pretty stable over time. So by the end of 12, 13 days, you can see they're pretty complete. In contrast, this is now the movie for the glutamate-treated cells. So glutamate T equals 0. So the neurites begin to form pretty normally during the first 3 days. But as the experiment proceeds, you'll see after 8, 9, 10 days, the cells become increasingly unhappy, and the neural network begins to break down. So you can always validate your observations by inspection of the individual images and the movies. Okay. To our surprise, when we did the same experiments with kainate, up front we saw slightly different observations. So kainate was capable of preventing neurite formation, and when we gave it on top of the established neurite network, we saw this very pronounced and rapid degradation of the neurites, which partially recovered. Only at higher concentrations it produces longer term effect on the neurite network. So we saw, made different observations with glutamate and kainate, which we thought was quite interesting. When we did those experiments, or the network disruption experiment with kainic acid in the presence of CNQX, we could completely inhibit the effect of kainic acid, as demonstrated on this particular slide. And interestingly, when we took the effect of kainic acid and studied it in the presence of BDNF, we could partially impair the effect at the later time points of kainic acid. But when we did the experiment in the presence of the proteinase C inhibitor H1152, we found an augmentation of the kainic acid induced destruction of the neural network. These data are summarized on this bar chart, where with normalization to the vehicle controls and defining the effect of kainic acid as 100%, what we observed was that CNQX by itself had no effect on the early and the later time points in the assay, the neurite formation, the neurite formation and neurite stabilization. Kainic acid ablated this, and if we looked at separated the post kainic acid treatment into 2 phases, the early and late phase, each of which could be fully reversed or inhibited by CNQX. BDNF in its own right augmented the neurite formation in these eye cell neurons, had no effect on the kainic acid immediate response, but the later phase response could be partially inhibited. And interestingly, H1152 had no effect in its own right on network development, but augmented the excitotoxic effect of kainate acid applied after 3 days. So some really, I think, interesting, perhaps unexpected observations. We don't fully understand these things as yet, but they give us a really good point of reference to begin bringing in other methodologies to answer these questions about exactly what's going on in these network cultures. Okay, so come to the wrap-up. Very conscious that everything that we show currently is with monocultures, there's a very well understood and documented role of support cells, astrocytes, and microglia in these types of model systems. These are quite challenging to include and monitor under phase, so we've also been working on parallel live cell non-perturbing methods for visualizing and quantifying neurons in the presence of astrocytes or other support cells. I show you just a couple of images here of fluorescent channel measurements of E18 rat neurons co-cultured with astrocytes, and in this case we've used lentiviral vector technology to introduce non-perturbing green in the left and red fluorescent proteins in the right to these neurons, and then we can use software tools to quantify the neural network through the fluorescence channel. Interestingly, there's really very little cytoplasm in these very fine neurites, so we've been using a neuronal membrane-directed tag GF2 in order to be able to clearly visualize the neurites using this approach. So this is a kind of watch this space example. It's something that we're working on, and in due course we believe we'll have more to offer those people using our technology. Okay, so we're almost there. Let me just summarize what I've shown you about the features and the added value of this live content imaging and NeuroTrack approach. I've shown you that it's label-free, involves no washing, it's image-based measurements of neurite outgrowth. These things are amenable to 96 and 384 well plates, and we've tested everything but the neuronal cell lines, and the primary and iPS-derived neurons that we've tested have thus far all worked very well in this system. All of the analysis that I showed you are embedded algorithms. They're relatively easy to understand and apply and cross-check and validate, and they provide full temporal tracking on the in the course of the experiment, so you can really watch these things unfold in real time. These measurements can be duplexed with fluorescent markers. I showed you YoPro3, some tag GFP, RFP. I also mentioned a caspase-3 substrate, which can be used jointly with the NeuroTrack approach. The added value of this approach, hopefully reasonably clear. The longer-term kinetics enable a far greater understanding of the biology and the experimental time course of the biology. We really believe that in comparison to endpoint measures, an awful lot more valuable information in this approach. The ability to run forward and reverse biology assays, I mean by that, for example, something like neurite formation versus form or breaking down established networks. So again, running these types of measurements would be really quite challenging using endpoint approaches. The ability to validate your quantification by inspection of the images and movies is very powerful. And another point which I really believe in for working with expensive and precious cells is the constant monitoring, visualizing of cells that you plate really helps you to do better troubleshooting and improved housekeeping of those cells, so you can get more data and more useful data, fewer plate failures, fewer optimization experiments are needed in order to really get you to where you need to be. Obviously, with a label-free approach, you don't need to buy expensive antibody labeling reagents, and the experiment protocols are really straightforward. There's no fixing, washing, or labeling steps that you need to consider. For NeuroTrack, you simply plate your cells and put them in the INQ site, as I explained. The applications in the neuroscience field are not restricted to neurite outgrowth. I don't expect you to be able to read the individual citations here, but there are in the region of 250 so published manuscripts now with live content imaging in INQ site. A relatively small but very recent number of those are in the neuroscience space. There's about 32 as of today publications in the neuroscience field. They span not just these neurite outgrowth measurements, but also other things listed on here: neurotoxicity, neural stem cell migration using a scratch wound module that we have in the absence and presence of biomatrices, And if you're interested in the process of differentiating somatic cells into pluripotent stem cells, then our whole well imaging platform and suite of software tools we think is really powerful and enabling in that regard as well. So I would encourage you to take a look at that. This full list of citations is available on our website if you really would like to drill in and look at how others have been using this technology. The final slide, before I hand back over to Jason, is just a list of where to go to for more information. Our website has most of what I've shared today. There's a full application note describing some of the methodology and the scientific data. We've presented some of the data I've showed you today at 2 recent scientific meetings. If you'd like a copy of those posters, we would be very happy to send them on to you. And I mentioned the list of IncuCite publications, which is again on our website. If you have any desire to move forward and either demo our technology or just find out some more about the instrumentation, the software, the consumables, then please connect through us. The simplest way is through our sales@essenbio.com mailbox. The other option that we provide for you through our biology teams here in the UK and in the US is a contract research outsourcing option. We have discovery services teams on both sites who would be very willing to work with you and assemble some study designs and costings for doing experiments on your behalf, and providing a solution for you in that way. On that note, I will wrap up and thank you for your attention, and we'll see what questions we've got. So I'll just hand back to Jason for a moment.
Thanks, Del. And yes, I want to remind everybody that if you have any questions or comments, you can type them in the chat box in the lower left-hand corner of your screen. And again, I want to remind you that no one else can see any of the comments that you type there. It'll just be seen by us, and we'll try to go through and get to as many questions as we can.
I have a list of questions here that people have submitted. As Jason said, I encourage you to submit more, and we'll just stay on the line for a few minutes and answer as many of these as we can do. First question: is the imager capable of detecting 2 neurites running in parallel? When we do immunostaining of neurons grown in culture, we find that the neurites often run parallel next to other neurites. Yeah, very good question. Thank you very much. We do see that, and we could go back and review that movie. We've called this phenomenon cabling, where parallel neurites try to almost wrap around each other, providing they are sufficiently distinct. If they're right on top of each other, then the software would not be able to discern that as more than one neurite. But even if they're very close to each other, again, I think we could probably go back and look at the movies to show that we can see that for the most part. It isn't perfect, and as I said, if they're fully on top of each other, we would struggle to resolve that. I suspect that would be the same in fluorescence, to be honest with you. I think the phase image processing has sufficient or probably comparable resolution to the fluorescence imaging resolution for these types of structures. Can you remove a scratch track manually? Yeah, good question. I'm not sure that you can. On the TPP plates that I mentioned, we see very few, if any, scratches. We typically run multiple images per well, and often multiple wells per plate for the same treatment group. So our solution to that would be to exclude a whole image from the analysis if we felt that the image quantification was compromised by an artifact arising from a scratch or any other imperfection. But I'm not sure in the software that you can manually remove a scratch. I will double check that for you and reply by email afterwards. recommended eye cell neuron plating density in 96 well for observing effects of neurite outgrowth inhibitor compounds. I should know the answer to that. I believe it's in the region of 25 to 30,000 cells per well. We don't typically, in fact, it's probably slightly lower than that. In fact, the slide I showed you was 96 well data, so the range that was on that slide, as I recall, was 8 to 20k per well, maybe 25k per well. So within that range, I will double check and get back to you. So thank you for that question. Do these human IPS neurons have the characteristics of mature neurons? A very good question, and arguably the $64,000 question. I understand from CDI that their eye cell neurons are heterogeneous. They're roughly 50% glutamatergic and 50% GABAergic. I don't think CDI argue that they have all of the characteristics of fully mature neurons, and I think this is the work in progress, that there's an increasing data set demonstrating that they have some or many of the characteristics of neurons, but my understanding is that they're somewhere between a immature and mature phenotype, and they're certainly not regional specific. And I think going forward we would really like to have, as I'm sure the community also would have, regional specific neurons. So for example, dopaminergic neurons or cholinergic neurons or forebrain neurons or whatever it would be. Is Essen possibly interested in fee-for-service work? Absolutely. From my last point on the last slide, please contact us. In fact, I'll contact you directly. Thank you for the question. We certainly are very happy to work in that mode, and we'll come back to you. Can the software count the number of neurites per cell body? Well, the software can count the total number of neurites per total number of cell body clusters. And that's not quite the same as the question that you ask, because if two cell bodies are in very close proximity, as is the case, and they do seem to like to cluster together, then the NeuroTrack module in phase cannot resolve those two cell bodies next to each other. It simply counts them as one cell body cluster. Now, the cell body cluster will typically have twice the area of two individual cell bodies, assuming that those neurons are not right on top of each other. So if you normalize the cell body cluster area, that's a reasonably good barometer of the total number of cell bodies in the field of view, and therefore a better approximation than cell body count, because cell body count, of course, doesn't take into account the number of cells that may be within each cell body cluster. There is another approach that we've taken to this which works, and we've shown it to work pretty well in immortalized cells which are easier to transduce and transfect, is we use nuclear targeted non-perturbing GFP and RFP probes to label the nuclei of cells in a non-perturbing way, and then we can count the number of nuclei, which we can differentiate from each other even if cells are in close proximity. So then we do a true cell count as a function of time, and then can normalize the total number of neurites per cell count. But that's a bit more challenging, and we've yet to do that in primary neurons or with IPS-derived neurons. Okay, so I think that's just about exhausted all of the questions that have come through on the moderator tool. So I thank you everybody for those. I will do my best to follow them up individually with you by email with more information where I wasn't able to provide it. So thank you very much again, and I shall hand you back to Jason again.
Okay, thanks, Del. We'll go ahead and keep this open for a few more minutes. If you have any other questions that come to mind, feel free to go ahead and type them in, and we'll be sure to answer as many as we can for the next few minutes or so. And as Del mentioned, if you have any questions outside of this webinar, you can feel free to email us at sales@essenbio.com, or give us a call on the number at on your screen. And of course, visit our website as well for all the HD images and videos, not only for NeuroTrack, but the rest of our cell player kinetic assays, and more information on the IncuSight Zoom live content imaging system. I also want to encourage you to connect with us on Facebook, Twitter, and LinkedIn. You can often find content there that you won't find anywhere else, especially special notices for events like these webinars and other things. So connect with us there for sure. Okay, it looks like that's it for questions for today. Then I want to thank you very much on behalf of Dr. Del Trezise over there in the UK. I want to thank you all very much for joining us today. And again, if you have any other questions, feel free to email us or visit our website, and we will look forward to seeing you next time.